Dr. Ebert,
Yes.
I sent you two emails, but I sent you one last week and the other one is this week, and
Okay.
you didn't respond to it.
Okay. I saw the one yesterday, and I need to get back to it, but I guess I missed the one last week. But yes, we can actually meet a little bit after class time if you want, because I saw that. I just didn't get a chance to respond yesterday. It's been a hectic semester. Okay.
Or if we can schedule another one because I have a class after
Okay.
like that class.
Yep. We might be able to schedule later today or sometime. So, okay. Okay. So let me show you guys what I'm talking about first with this other mini exam, because what I thought is I'm not just randomly adding more work, because that would be wrong, but I know some people have been concerned about some of the exam grades, so I wanted to add another thing that should be, it's kind of in between an exam and in between homework. It'll be timed, so it's a little bit more like an exam there, but it's very open-ended, and as long as you kind of read, it'll be more like homework in that regards. I just want to see that that you assimilate information. So to that end, I think I had shown that NORD, National Organization of Rare Diseases or something. Probably would help if I hadn't minimized my screen. Right? Okay, let's open it back up there again, and then find you guys and share screen. All right. So if we actually go into assignments, what you guys are going to see sitting up here now, well, at least I'm going to give you my view. You will see eventually, besides the homeworks, this potassium channel test and exam number 5. Okay, so exam number 5 is going to show up on Tuesday, and then I'm not sure about this one. I don't want to give anything that's after Thanksgiving, over Thanksgiving, but I also know that you guys likely have exams next week for other classes, so that's why I'll kind of do a little poll about that as well too. But this thing will show up at a time that we all agree on, and it'll be total time. I wouldn't expect it to take more than an hour. Let me say 2 hours including prep time, but it should only take like an hour including prep time. All right, and then once Canvas loads my modules, clearly I have too many of those. And I sent this out yesterday. There's this link to the KCNB1 encephalopathy, which is this very rare disease that only affects about 65 people. Of course, the disease has only been known since somewhere between 2014, 2017, so it's fairly new. Oh, that reminds me. I forgot the other question I was going to ask in there. I got one more question to add. But there are also 2 papers that involve it. One, the primary paper that describes it is from 2017. I think the disease was really first discovered in 2014 as an offshoot of some other infant epilepsy type diseases that were in there. But this one affects a specific ion channel, which is what we're going to talk about in class at the start today. With our regular lecture, this kind of describes it. There's another one here that talks about a bunch of mutations, because there's not just one mutation that results in defects in this particular. It's a family of potassium channels. Do you need to read both these papers? No. In fact, this second one is actually very dense. And in fact, let me show you what it looks like here. So let me stop sharing this and have those open. All right. So this. Okay. Yeah. I forget. Yeah. Okay. That's one nice thing about the Adobe Cloud is that it shows you a bunch of stuff that's up there. All right. So this is the second paper, because nope. Sorry. This is the second paper, the one with all the mutations in it. All right. And it is, it's an awesome paper. It's really, really good, but it's very dense. So if you find this type of thing interesting, this might be something worth digging into. We've got the abstract in here. That's not too bad. Materials and methods. They talk about the plasmids they use, so you can see some examples of these things we talked about in the previous material. Patch clamp recording. I don't know if anybody has taken classes or worked with Dr. Overholt, but I believe he's done some patch clamp recording. And I think there's one or two other people in biology who do that as well. Talk about cell surface bitinylation. Okay, yeah, we're cool here. We're cool. Flow cytometry, data analysis, and it just keeps going. We get to our results. This is actually kind of neat. We're looking at homology across organisms. You can see here that we have several transmembrane regions. I believe we have what, 1, 2, 3, 4, 5, 6 transmembrane regions. Okay, yeah. So that's a little bit different. A lot of channels will have 7 or 12. So this one's got 6 and some other stuff in here for this pore helix. This is kind of neat. Um, and then you can see where these mutations are found. Okay. And then here's the general crystal structure of this molecule as well too. So far, it's not too bad, but this paper goes on and on and on, and we start seeing, various mutations. And you can see here we have an arginine that gets mutated to a cysteine, or a threonine that goes to methionine. And there, it turns out, there's a huge number of these mutations that all result in a defect in here. So, what this tells me is that, these 65 cases that have been found, um, you know, there's maybe like 3 or 4 mutations for each one of those, or 3 or 4 patients for each one of those mutations. So that's not a lot. So again, this is a very rare type of disease in here. But again, this figure starts to get crazy, and here we start looking at the patch clamp recording, the current traces in here, for mixtures of these as well too. And it just goes into way flow cytometry, way more detail than we really need or want at this point, unless you're actually going to go into this area or look at this type of technique. So like I say, it's fascinating, because it's such a rare disease and apparently such a critical gene. This KV2-1 family of potassium channels is very very important, obviously, because anybody who has a mutation in there, any one of these types of mutations tends to have some pretty severe effects in here. Okay. And there's yet another paper that's come out about this. The original paper kind of describing it is maybe a little simpler, and this would probably be more accessible to read. And they talk about I think they have like a half a dozen cases in here. Actually, that's closer to 10, I guess. Nine cases. They split them up into some cohorts there to look at them. And they talk about, this is the thing I thought that was kind of neat. The onset of the disease. You can see these are all babies. There's one, that didn't start until 18 months, but everybody else is before a year old. And they start with seizures and spasms and kind of, all sorts of things that are in there. Is really kind of fascinating. To look at a couple of these patients, because you can see most of these are young. Only one of them is an adult. One of them is 17. And in the study, this 22-year-old female was actually able to graduate high school with with some assistance for, like, was it a ISE, or individual, let's see, IEP, individual educational plan, or something like that, but made it through grade school, okay. So it was really, her case wasn't as severe, as the others, but you can read about some of the effects in here. Okay. Pardon me. What do we have here? All right, but yeah. Anyway, way more information than you really need. Again, we can see this, a non-color version of this gene in there. Really, I'd incorporate that picture in the question, but I don't want to make this long. I don't want to make this long. Okay, but anyway, that's a lot of papers in there. But the only thing you really need to read is that link to the NORD site, which is much more accessible, much easier to read. Like I say, I've got this exam set up, and it's like 5 or 6 questions, so, and they're not terribly long or bad. Um, and most of them are asking you, okay, so what do you think about the why would you expect to see this? Or I give you some of the information in there, and say, okay, so we have maybe this particular mutation, and it's different effects from this mutation, why. So I just kind of want you to theorize and kind of put together some of the stuff that we've been talking about over the semester to look at that. And then there's a couple questions just about ion channels in particular. So like I say, I'll have a little survey or something come out there later today. Once I get my brain functioning, and then we'll work on deciding exactly how to do all that. Okay. So I probably talked too much about that, but I'm enamored of that. It's like, oh, something new and exciting. So it'll be a kind of fun, hopefully, test. Not maybe not the greatest excitement for you guys, since it's, oh no, it's another exam type thing. But again, my whole point behind this is just to have you guys, use the information that we've been discussing and maybe apply it a little bit, and make it in a format that will contribute to exam grades, so that overall exam grades will probably be impacted up a little bit, because it's not going to be a terribly hard exam, but I just want to see how you think. Whew. I was gonna say, yeah, if you worry about it, like if I haven't absorbed anything, if I've learned nothing this semester, maybe it'll be a hard exam, but I don't think it'll be too awful. Okay, but back to the last of the material, and like I say, this KC NB1 mini exam deals with ion channels. So let's talk about ion channels a little bit. Okay. So we had talked about previously our G protein coupled receptors, and we talked very briefly about tyrosine kinase receptors, all right, and looked at things that are associated with them, the way they function, and all that. So we've got 3 more types of receptors to talk about for signaling, and so we're going to talk about ion channels to start with today. All right. So our ion channels transport ions across the cell membranes, and this can be either out of the cell or into the cell, or bidirectional. And basically, if we look at a gated ion channel, it can respond to either a change in the membrane potential. If we keep pumping ions out, we're going to change the charge difference between the inside and the outside, and eventually our electric potential will get high. And when that potential hits a certain point, we have like a potential sensor that's part of the ion channel, and it will sense that increase in voltage and open up the channel and allow ions to flow through, and thus allowing the membrane to depolarize. So when it depolarizes, the lid will close. Right? So that would be a voltage gated ion channel. We can also have a ligand gated ion channel, and I'm tempted. I'm going to use a blueberry. All right, so we've got our channel that's closed. Our blueberry comes and binds to its binding site and opens up the channel, allows them to flow through. And then Dr. Ebert eats the blueberry, and the channel closes again. Sorry, very late breakfast too. All right. Plus, I have to eat the blueberries while my daughter is at daycare, because she will eat all the fruit in the house, all the fruit. All right, but those are our two cases. We can either have membrane potential causing changes, or we can have something bind that causes the changes in there. And our ion channels, we find a lot in the nervous system. Okay, so we can think of our neural activity is a lot of electrical activity. So there's current flow and voltage polarization and depolarization that's happening in the nervous system. So that's why we see a lot of ion channels that are found in neurons and in other nerve cells and associated cells with those. And we're going to look at an example of a signal transduction down a nerve pathway involving both voltage gated and ligand gated. Okay, so we'll often see like some voltage gated sodium channels, a nicotinic acetylcholine receptor, glutamate receptors, GABA receptors in the brain. All these things are involved with ion channels. Okay. So if we talk about polarization, the inside of the cell is usually more negatively charged than the outside of the cell. All right. So we typically see a voltage drop across the membrane of 50 to 70 millivolts. All right. And this is largely due to this asymmetric transport of sodium and potassium ions. So maybe we kick out, um, um, now of course I got the blueberry skin stuck to the roof of my mouth. All right, we kick out 3 sodiums and allow in 2 potassiums. All right, so the net effect here for this antiporter is that we are kicking a positive charge out. So we're making the outside more positive and the inside more negative. And as we do that, we build up that electric potential. We also can get a chemical gradient there, but we'll see that increase in potential as well too. Okay. So if we look at an example of this, here is our antiporter. We let 2 potassiums in, kick out 3 sodiums, and we're going to see the outside of our membrane is going to be more positively charged. Okay, this can be this particular antiporter maybe requires ATP to do that. Okay, and then we can see typically in the cell. We're going to see sodium will move down a channel to flow into the cell. Same thing with calcium will flow in. Potassium is flowing out. We tend to have more potassium in the cell, right? And chlorine will tend to flow out against a concentration gradient, all right? But we've got this negative charge out here, this positive charge out here, and chlorine has a negative charge, so it's actually flowing down a voltage gradient. So that's why it's favorable for it to flow to higher concentrations. This is our typical healthy cell. Our typical concentrations of sodium and calcium are higher outside, and potassium and chlorine are higher inside. And again, we have multiple channels allowing things in and out that are all ideally synchronously regulated. Okay, so let's look at an example of a couple ion channels in nerve signaling. Okay, so if I poke myself in the finger, I get a nerve impulse. I can feel that it travels up there. And basically, this propagation impulse is going to involve voltage-gated sodium channels and voltage-gated calcium channels that will then allow the release of acetylcholine, which opens a ligand-gated channel on the receiving cell, and then this continues down here. So rather than me trying to explain that in words, especially when I've got a pounding headache, we have this awesome picture here. Okay, so here we get some sort of signal, and we're like, oh no, a voltage, something happens, I poke my finger or whatever in there. So my voltage-gated sodium channel is going to open and allow this influx of sodium. Okay, so as that happens, we're going to see potassium flow out to repolarize the cell. All right, so this flows in, depolarizes, it's going to flow out. But what this means is we're going to have this initial gradient of pardon me, of sodium. Oof, man. All right. So when that happens with this depolarization event, when we depolarize, that's going to open up the calcium channels and allow calcium to flow in. All right. So our calcium flows in then, and when our calcium flows in, it's going to actually trigger the release of these secretory vesicles. So we're going to have exocytosis of these little vesicles that contain acetylcholine. Okay, so high calcium levels cause them to move to the cell surface near the cleft here and release acetylcholine. Okay, and then acetylcholine, when it's released, will flow across the synaptic cleft and bind to a ligand-gated channel that will allow in sodium and calcium and propagate this to the next cell. Okay, so we basically have the signal that comes in here. We release acetylcholine and it transfers the signal to the next cell, and then it goes to the next cell and the next cell and the next cell until it gets to the muscles. And I twitch or poking my finger like, oh, I pull my finger away. It's like I tell the doctor it hurts when I do this, and he says, well, don't do that. Jokes from the 1950s, maybe 40s, 30s, who knows. All right. So hopefully that's not too bad. And what we really have is a pulse, and I've been looking for it and I can't find it. But somewhere when I was a grad student, so it's been a long time, there was actually like a little film grainy film, because it was back in the 1920s now. A grainy film where you could actually see these gradients traveling in the cell. And it's like little waves of calcium. So a couple things to note, we're kind of drawing these like, oh, we put all the sodium ones over here and all the potassium ones over here, and all the sodium's in the north side and the calcium's in the south side. But these are really pretty evenly distributed around the cell. So we're constantly, they're just side by side. We're just drawing them this way to try and make it a little clearer to see. All right. But what we have is this flash influx of calcium primarily. You can kind of follow calcium in there, and then as soon as the calcium gets in and triggers this release of acetylcholine, you've got things like calmodulin and calsequestrin that will, and another thing that it'll absorb the calcium, transport it out, and then it'll flash back in. So really, this nerve signal, you get these little waves. It's kind of like waves in the ocean. You get these little waves of ions flowing in and out, flowing in and out. So it's like um, depolarization, repolarization, depolarization, repolarization, and this is happening really, really quickly. So again, that's probably why we have acetylcholine in secretory vesicles, so they can be, oh, okay, release, oh, release, oh, release. So this is a very fast process. Okay, so once our acetylcholine is released and it binds to our ligand gated channel that allows in sodium and calcium to the next cell, again, this kind of same thing happens. We see, hey, this influx of charge results in depolarization. So we see this in, and then we start pumping out our potassium, we get repolarization, and this just continues on and over and over. So there's a huge amount of electrical activity that's taking place within the nervous system, and that's critical for all these signals to go back and forth. So if you mess with that, like say make a mutation in a potassium channel, then you mess with the way that the nervous system functions. Okay, all right. So hopefully that's not too bad. I do want to kind of just show you again. I'm sure you guys have all seen like the stuff at the bottom in here, and the notes on the slides of the cell. I particularly like these notes for this figure in here because it does go into some pretty good detail. So that's I find helpful. So hopefully that's not too bad for ion channels. Okay. All right. So that's 3 out of 5, right? Next one, this is a short one, and this is a shame. This is where if this were a more normal semester, I would have more slides and more discussion about integrins in here. Integrins mediate cell adhesion. They're one of my favorite signaling molecules because they're so simple. Also, they may be involved in the research that I'm involved with, but we're not sure about that yet. But integrins on one slide basically are basically little telephone poles that extend. So if my elbows are the outside of the cell and my hands are the inside of the cell, integrins are heterodimers. So we basically got 2 proteins. There's an alpha type and a beta type in there. And they're attached on the outside to extracellular matrix proteins. That's why we have to have these particular sequences like arginine, glycine, aspartate. That will allow them to bind to things like collagen, fibronectin, various other extracellular matrix proteins, right? And then on the inside, they're attached to the cytoskeleton. So if I got my random wires hanging around here, the inside is attached to the cytoskeleton. So if we have something that happens in the extracellular matrix, we exert tension in here, and we can cause a conformational change in the integrins, and then that conformational change will pull in the cytoskeleton, and that transfers the signal across. Same way, if the cytoskeleton changes, we can transfer that signal back. So I always think of it as like the old West telegraph poles or something like that, where you're sending this a little signal going there. But typically, our signal is going to come from the extracellular matrix, go in, cause these integrins to move, which is going to rearrange the cytoskeleton a little bit, and that can promote gene expression. We can turn off certain genes, turn on other genes in there. All right. All right. And then newly expressed genes will often bind to the intracellular domain of them. which can trigger the extracellular response as well too. So basically, say, hey, something's happening over here. We're going to make some new gene proteins, gene products on the inside in response to that, and that's going to tell the outside, the cell what to do regarding whatever is going on outside. So whether it's maybe inflammation or some type of growth necessary, like for example, if you think of like when you get a cut and your body is going to heal a cut, you got to regular regrow some connections in there. That physical damage may have an integrin mediated response to that. All right. So those are integrins. And that's number 4. That's one slide in there too. So the big key is I may try to put another slide in here or something, another picture, just so you can see this heterodimer interaction. That's going to you pull on one side, you get the response in there. Maybe a better thing would be like the old time bell with a bell pull in there. You got a rope that's hanging down. I guess church steeple. We'll do that. You pull on the rope and up through several floors, it rings the bell. So that's essentially what we've got going on here. And that's number 4. So we've got one more type, right? All right, so our last type is going to be our hormonal regulation, where we're going to look at the nucleus, okay? So here we may have hormones that are released and they will cross the membrane, both the cell membrane and the nuclear envelope, and then they will go and bind to a nuclear receptor. And when they bind to the nuclear receptor, that will trigger changes in gene expression. Okay, so maybe we have some particular hormone. Keep in mind, most hormones tend to be hydrophobic, so they can cross membranes fairly easily. So that's why in this first case, we have maybe some type of serum binding domain that will carry this through the bloodstream, since the hormones don't diffuse very well. It's not the most efficient, but keep in mind too that a very tiny, tiny concentration of hormones can have a huge response. Um, All right, so we get to, when we transfer our hormone from wherever it's manufactured, and I think we talked about, the whether they're locally acting or throughout the entire body, and the terminology for all that. But they get carried there, um, you know, um, released to bind to flow across the membrane into the um, nucleus, and then they can bind to their receptor. And when they bind to the receptor, they're going to, of course, induce a conformational change that will either make it more likely to bind to the DNA or less likely to bind to the DNA. But we're going to look at this one in case and say, hey, this is going to turn on a gene. Okay, so it binds there, makes it more likely to bind, or it makes it more likely to form a complex, or less likely to make a complex. We got some pictures coming up here. In any event, whatever the hormone binds to in the nucleus, its nuclear receptor, is going to induce transcription of new mRNAs and lead to a new protein in here, which can change the function of the cells. Okay. All right. Now, I think if I remember correctly here, no, did I really not? Oh, I took those out. Damn it. Okay. Yeah, I did have some really cool stuff. I must have been on a binge of deleting stuff in here. So I guess our hormones are in one one slide. That's terrible. So what happens when Dr. Ebert had a pounding headache all morning? All right. Well, anyway, and maybe it was in one of the maybe it was in chapter 11, but I know there's a place where you could see that we could have these hormones bind and turn things on or turn things off, or make them more likely to bind or less likely to bind. But the important fact here is for our signal transduction, we're actually crossing both the cell membrane and the nuclear membrane and going directly to the nucleus to induce transcriptional effects. Okay. So, we may have again, if this is our nuclear receptor for our hormone, it can interact with a co-activator or a co-repressor and alter the transcription of genes. Right. So you can think of these co-repressors, co-activators as like Darth Vader. It's like, I have altered the expression of the gene. Pray I don't alter it any further. You got to have a Star Wars quote to end the semester, right? I was like, Dr. Ebert's such a dork. I can see my evaluations now. All right. So those are the five types of our signal transduction mechanisms. Obviously, when it comes time to prepare for the exam, you might see, one question on hormonal regulation and one question on integrins, and maybe, well, probably one or two questions on ion channels. But we're going to have our own little mini exam on a particular KCBN1. So the bulk of the things we'll be talking about will be the tyrosine kinases and the G protein coupled receptor. Right. Excuse me. All right. So this brings us to kind of just like wrapping up this semester. There's some stuff in here that this is more application oriented. Okay. So we can look at coupling our various receptors with enzymes. Okay. So in this case, we can talk about chemotaxis. All right. So if, for example, how does a bacteria know to move towards a food source? It's like, and this results on chemotaxis. So basically, our bacteria have our little flagella down there. They're going to spin around and they're going to, drive them in a direction. What direction do they go? They're like drunk people in bumper cars. They're just going to go in random directions in here. So what'll happen is, we'll turn this on and we'll go in a direction for a while, and then they'll just tumble. They'll just kind of sit around there. It's like they're scanning. It's like any food here, any food here, any food here, any food here. If they don't see any, then they'll just drive in a certain direction for a little bit, and that's our run. And then we'll kind of tumble. All right, and it'll say any food here, any food here. Basically, if we look at our food source for bacteria, we'll call that our attractant. Maybe there's some that's located over here. So, the darker the color, the more attractant we have. So it says, hey, there's a little bit of food over this direction. So they sense some in that way. So it's going to, run over here and it's going to tumble around and say, oh, there's kind of food all around us. I'll go this way a little bit, and then tumble around and say, whoa, there's less food over here. So I'm going to, and eventually, through this kind of random action of tumbling and running, they will move towards the higher concentration of the chemoattractant. Okay, and this is all regulated, okay, through a phosphorylation cascade in here. We have this you know a receptor that's bound to a His kinase. So it'll get phosphorylated by ADP. When that's phosphorylated, it'll phosphorylate this response regulator. When that becomes phosphorylated, it will move to the flagellum and basically turn that on and drive it in a direction. So basically, we say, hey, chemoattractant in this direction, we're going to turn on the motor and go that way. And then, of course, this adapts. We see that adaptation or desensitization. Our response regulator becomes dephosphorylated, and that's when we do the tumbling again. And then, if we have one particular direction where you see a lot of chemoattractants, we're going to, activate that motor protein again. Okay, so that's why bacteria can find your food. Yeah. All right. I don't even want to talk about cell phase and stuff like that. I think you guys have enough. But the cell cycle is regulated by cyclin-dependent protein kinases. So we have these various cyclins that are elevated or reduced in concentration based on the particular phase of the cell. So you can see the G1 to the S phase, cyclin E is elevated to maybe initiate the S phase, and then it kind of falls off. And in the S phase, we see cyclin A that gets elevated all the way through G2. And at the end of G2, we turn on cyclin B and we turn off cyclin A to get our M phase, right? And then we start it all over again as those fall off, okay? I'm probably not going to ask anything about that, but again, these kinases again play a role. So phosphorylation, massively important for the typical regulation of activity in here. And this is just kind of introducing, our concept of regulation. Okay, so if we have no cyclin present, we're going to have this cyclin-dependent kinase that is inactive. Okay, we synthesize cyclin, and we're going to turn on this cyclin-dependent kinase. All right, so our so we might have cyclin bound here, but because this particular tyrosine is phosphorylated, it can bind, but it's still inactive because it's going to prevent the cyclin from being phosphorylated here. Okay. Um, so we can phosphorylate this threonine in the other side, which will activate this, you know, a huge amount. We can see that, we have this cyclin-dependent kinase is going to phosphorylate phosphatase, which activates more cyclin-dependent kinase. So we're going to see this massive increase in activity here. We dephosphorylate the tyrosine, phosphorylate the threonine. So that's some complex activity going on in there as well too, because usually our kinase events are kind of indiscriminate. So here we are being very, particular, specific about what we phosphorylate and dephosphorylate. Once this is phosphorylated and activated, we can see that we have this other protein that gets phosphorylated and activated. Anyway, but we're going to go through our particular phase, and then as we enter into that particular phase, we're like, way too much activity. So if we go back up a slide here, you can see, hey, my cyclin A is made way too much of it. So now it's time to die. We're going to have our DBRP protein getting phosphorylated, which is going to add ubiquitin to the cyclin. Ubiquitin is a signal to take it to the proteasome and degrade it. And when it does that, our cyclin comes off the cyclin-dependent kinase, and our cyclin-dependent kinase becomes inactive with no cyclin there, and our tyrosine becomes rephosphorylated, and our threonine becomes dephosphorylated. So that's a lot. And then cycle just goes over and over again. Okay. And of course, when you've got cyclin A, cyclin B, cyclin C, so on and so forth, all these different cyclins with their various cyclin-dependent kinases that flow during these cell phases. Okay. So the idea is during the cell cycle, we have variable regulation to kind of turn on and turn off these various productions of these cyclins. And we see this regulation is through a signal cascade. In this case, it's a kinase, primarily a kinase dependent signal cascade. But as we change things, we have regulation occurring. Okay. So, and this is just kind of like a little summary of that. What triggers all this? Maybe growth factors or various cytokines will trigger this phosphorylation cascade, and we activate these transcription factors to turn on our cyclins and cyclin-dependent kinases, and maybe our various enzymes for our DNA synthesis to get into our S phase and all that good stuff in here. Now, these are all good and necessary. And if everything works right, we get an organism that grows, cells divide, they split off, it gets bigger, and reproduces, so on and so forth. Everything is good, but things can go wrong. So we can see constitutive activation of cyclin-dependent kinases can lead to some forms of cancer. All right. So basically, your epithelium, if you think of that, the epithelium is constantly being replaced. All right. So we need our cyclin-dependent kinases to have those cells divide and produce new epithelial muscles in there. Okay, or sorry, new epithelial cells in there. As we progress, maybe these become a little bit more active, and we're growing too many of them. Okay, so you can get excess epithelial growth in here. All right. And then we get these basically small adenomas and large adenomas, basically these excess cells that are in there. Okay. The problem happens is if we continue to have these constitutively active, then we kind of turn them on. We lose some of these regulatory factors, and this uninhibited growth becomes uncontrolled, and we develop cancer cells that are constantly growing, constantly reproducing, and they can metastasize and spread to other areas as well too. Okay. So this whole process is kind of oncogenesis of growing too much. So we can see that we have these various growth factors in there. So what do we see here? Our epidermal growth factor and our COX-2 both promote this. And then our PGDH dehydrogenase and our tumor growth factor both kind of be inhibited here. But these factors get turned off, in particularly in colon cancer. So if you think of like polyps, like they always talk about the colonoscopy of the various presidents, whatever. It's like so and so had polyps removed or something like that. So basically, this uncontrolled growth, these polyps can be epithelial cells. If they're just growing out of control, you can just, was it benign? They can be removed. You can remove those cells. If they continue growing back, they can develop into colon cancer. All right. So, understanding these pathways can lead to treatments for cancer. So, maybe protein kinase inhibitors. If we can prevent some of these phosphorylation events from occurring, then we can prevent this growth that's going on in here. The key is we have to balance that because essentially, most cancer drugs are highly toxic and very poisonous. If you think the stereotype of cancer treatment is, all your hair falls out and you lose your appetite because fast growing cells are killed. As we understand more and more about different types of cancer, we can make better and better targeting proteins in here. Roscovatine. I had to think. I had forgotten this was in here. I think I actually used roscovatine as an inhibitor in my Schwann cells for something like that. But this is basically a protein kinase inhibitor in here, and it basically inhibits cell growth. So I use that as a negative control in my Schwann cells because I use a Schwann cell line, so it's immortalized. They're in essence cancer cells. So roscovatine as my negative control will prevent them from growing by inhibiting the kinase factors in there. And you can see it's got a close enough structure that it mimics ATP. It'll bind to the binding site in cyclin-dependent kinase 2 and prevent it from functioning. Okay, but it's just kind of a brief example. Look at one particular type of colon cancer from uncontrolled epithelial cell growth and possible ways to treat it. Okay. All right. Wrapping up, and again, this is one of these things where I wish I could go into excruciating detail because we're doing apoptosis in like one slide. So that is, I could easily give you a 60-minute presentation on apoptosis, although it's a few years out of date now. I need to update it. But basically, everything has a lifetime. All right. So you think, you look at majestic trees growing in the forest, but even trees reach a point where they're kind of done. There are a few weird exceptions to that. There are a couple like 1,000-year-old trees. There are fungi that can grow, and technically be like the largest organism. They're, still a single organism, but that's redefining. But trees have a life cycle. They live longer than us, but eventually they're going to die. So our cells have a life cycle as well too. So cells, if they live a long time, they accumulate mutations, and they become diseased. So one of the ways to prevent the whole organism disease is to have this programmed cell death or apoptosis. All right, and this is modified through the activity of caspases. Right, so there are a whole bunch of caspases, and they're named for the order they were discovered and not in the order they activate. So caspase 8, I believe, is the first one. Nope, sorry, I might be wrong. Might be, it's caspase 8. Yes, yeah, okay. So it's really kind of cool if you have a morbid sense of humor to study apoptosis because we talk about particular proteins and receptors that have death domains. So just like we mentioned last time about SH2 domains and SH3 domains and SOX and all that stuff, you can kind of mix and match. We have death domains, all right? And the death domains are going to interact with our caspases. And our caspases are actually found in the cell as procaspases, sometimes as pre-procaspases. So they're in an inactive form. And what will happen is maybe we can have a particular factor, like this is tumor necrosis factor alpha, TNF alpha. When it binds to its receptor, it will induce a conformational change on the inside that will allow the tumor response portion of the protein to bind procaspase 8, and then it cleaves off a section in there and activates it into caspase 8. All right, and then caspase 8 turns on a whole bunch of other caspases. So if you go back to like the ancient times when you had the watchman on the mountains, it's like, oh no, here comes the invading army. Let me light this fire. I forget, there's some like movie a couple years back that had this, so you light the watchfire, and you've got watchmen on other mountains, and they see that one fire, so they light their fires, and so you get this big signal here. That's our Caspase. Is like, oh no, it's time for us to die, basically here. So we activate all these Caspases. Okay, Caspase 8 also goes to the mitochondria, which is again why I like Caspase 8. It goes to the mitochondria, and it triggers the release of cytochrome C. So if you remember, cytochrome C is in the electron transport chain, and it just shuttles electrons back and forth between complex 3 and complex 4. If you have this sudden massive release of cytochrome C, it's going to activate this protein called APAF1, which is going to trigger the actions of the apoptosome. Basically, this is a whole big complex of protein machinery designed to neatly cut up and kill the entire cell. So again, we have procaspase 9, it gets activated in a caspase 9. All these caspases come together, and they start, systematically chopping up everything in the cell, and killing the cell very neatly, so that you can recycle and reuse different pieces and whatnot in there. So it's basically our programmed cell death. Okay, so maybe we're not quite as morbid as Dr. Ebert, and we don't like to talk about this and think about this, but we can look out the windows and see the leaves that are falling off the trees, or mostly have fallen at this point, thanks to that storm a couple days ago. But essentially, we have apoptosis that's being triggered in those leaves. The leaves are held onto their stems. Okay, so they're nice and bound in there. It's all nice and good. We have certain proteins that are involved in holding them on there. As we see reduced amount of sunlight, that lack of sunlight triggers apoptosis in there, which slowly starts cleaving these proteins in the cells that are holding the leaves on there, and we get programmed cell death. We weaken our connections, and eventually the leaves will just fall off, or if we get a storm, the connections are so weak that they'll just blow off very easily. So that's an example of programmed cell death. As the trees are going dormant for the lower concentration of sunlight in the wintertime, they don't need these leaves, which require a lot, and they produce a lot of energy, but they require a lot of energy too. So we're going to trigger apoptosis to get rid of them, so that the tree can go dormant. And in the springtime, with the increase in light, from as the light gets longer, then we will regrow new ones. All right. So that's this chapter. That's this semester, basically. But this chapter is primarily all about cell signaling, the different ways that we can signal cells, our G protein coupled receptor, our receptor tyrosine kinases, our tyrosine kinases. There's basically a bunch in there that we talked about. So if we look at the initial slides in here, those are very important. And where we're really going with all this is to get to regulation. So if we go back here to these early ones, our 5 features of the signal transduction system, and I think there are 5 types in here. Yeah, something like that. Okay, so yeah. So that's basically this chapter. Woo! Yeah, we made it, right? You guys can all say, woohoo, yay! All right. You guys, if I haven't put you to sleep today. That's why I'm coughing. It's like, you wake everybody up. No. All right. So with that said, let me share one more thing, because might not have expanded my slides, but I did post the study guide. All right. So I tried to make the study guide very big and very detailed. All right. So we've got the classes in here. If we go back to chapter 11, we can talk about, you definitely want to draw the membrane, but you'll have to be able to describe the lipid bilayer, integral versus peripheral proteins, loosely associated versus, integrated, glycosylated lipids in there. Maybe some differences in there, but you get a lot of that from biology anyway. Functions of the membrane. So looking at slide 9, I believe it's slide 9. To go back and check that to make sure. But all this stuff that's in there. And then, we get into, our snaps and snares, our FRAP, our flipase, flopase, and scramblase, some different experiments in here. I have a question that I love about FRAP. So understand FRAP. That'll be good in there too. It's like, I love that question way too much. And then, active and passive diffusion, looking at those types of transport across there, that'll be important as well too. And certainly, you'll see big questions about the different types of transporters. I think whether six or seven of the five, 6 or 7. So yeah, yeah. All right, and then now chapter 12. It's all much more straightforward here. Um, this is old. I think we've replaced JackStat with the KSR, so don't worry about knowing that. But insulin is a good example in there. But being able to find things like crosstalk and stuff in there. So adherence function, nuclear receptors, consistently active, and problems again are uncontrolled growth leads to cancer in there. So okay, all right, that's way too quick of a review, but that's what's in there. Okay, so and of course, oops, I'm getting messages from all over the place about my son's school now because they've had connection issues, unlike us. Yay! So yeah. Anyway, we have much fewer students. We have hundreds of thousands fewer. Okay. So what I want to ask you all now, I have a question for you. And I know everybody's shy to talk and nobody's on camera or anything like that. But if you would prefer your final exam to be mostly multiple choice, just give me a little thumbs up by your name in there. And I don't know if you kind of click on a little more. No, that's not it. There's a way to do that. Somewhere down here at the bottom. Reactions at the bottom. Yeah, you can just put like a little thumbs up there. Yay! So how do I undo that? It's a good question, isn't it?
Dr. Ebert,
Yes.
if we were to have our whole exam be multiple choice, would that mean more multiple choice questions, like 50, say short answer 25?
Yes. So here's my thought. Um, so if it would be mostly multiple choice, so we're talking like 33 to somewhere in the mid 30 to 40 multiple choice, and like a handful, like 2 or 3 thought questions in there for the exam versus like 10 to 15 multiple choice questions and more essay questions, like 10 or so. Okay. So it kind of comes down to what you prefer. Um, and I'll let you guys that are here kind of vote for that because I'm asking the same things. I'm just asking them in different ways. So if it's the mostly multiple choice, like the multiple choice questions will be worth about the same, about like two to 2.3 points each. So yeah, they wouldn't kill you. It's just it would be more essay questions versus less essay questions. As opposed, basically kind of like as opposed to explain G protein coupled receptors, it would be multiple choice question about the alpha subunit, and multiple choice question about the say like second messenger, and a multiple choice subunit about how desensitization and stuff like that. So rather, yeah, okay. So yeah, I see. I don't know how well this will work here. I may have to actually make this into like a little poll question as well too, that I send out to the whole class. But my impression is that in general, more students would kind of prefer multiple choice. Not to say that that's a crutch, but people students tend to want more multiple choice because the right answer is going to be there somewhere. I can make all my questions or none of the above. No, I'm not that mean. I swear they won't be that bad. I try and make them straightforward, but some of them are hard. In tests, just this particular test. This particular test is pretty middle of the road for this semester. It's definitely much less challenging than exam 2, but it's not as easy as exam 3. So it's kind of in between there. I think this is usually like the second highest grade historically, with that one weird exception for where I was going for 3 weeks and then the students took the exam. That was a bad scene, which is why I want to do it on Tuesday so that you guys don't forget stuff. All right. All right. Well, I'll make a little poll question about that and post that very soon because I know some of you have classes to go to, and I have a meeting in about 15 minutes. So, busy day. All right. Okay. And just a note, while I am doing the recording here, just a note.